most of us, I think all three of us did get our start in the wet lab on the bench. And you typically would start exactly where you just outlined. I want to target a protein. It's an enzyme, it's a receptor, whatever it is. Let me look at the structure. Let me use medicinal chemistry to target a molecule. Oftentimes you have large libraries. It goes through the sequence. And the numbers are very disheartening. I mean, you threw them out there, Jack, but we're all used to hearing this, right? Approximately 10 years to develop a drug. I mean, once you know the target, even at that point. And less than 10% success rate. I know I've been guilty of becoming complacent with that. You try to shave a year off, 2 years off, but I think we're at a point with technology where we can do much better. So can you help us to understand what are we doing with AI right now that can shorten the drug development timeline? Like, what should people be expecting today? And then I'll ask you about 10 years.
Sure. Let me start, and Nadia, I'm sure has other examples, but when you look at the end-to-end of drug development, drug discovery, and then development, and also in the clinic, it's absolutely critical now to think about AI all throughout the process. Target identification validation, as you mentioned, can take years on its own, and that's an area, and I'll ask Nadia to delve into that. Once you have the target, you want to drug it either with a small molecule or increasingly, a lot of us are interested in biologics, these larger molecules. We're having a lot of success there. Keytruda and other checkpoint inhibitors are an example of a biologic that has literally saved the lives of millions of people out there. And then in clinic itself, people sometimes think, AI, its job is just to deliver it to the clinic and then we're done. Actually, what we're seeing is when something is in clinical trial, we find that of the failure, half is due to lack of efficacy and half is due to severe side effects. We call them SAEs, as you know, all too well, Shoshana, from your backgrounds as well, severe adverse effects. And often those drugs then go on the shelf, they get killed off, but actually we can now save some of those drugs. We can analyze what is that adverse effect or that SAE, and we can understand what part of the cohort of people in that trial are being impacted by that interaction. It's not everybody. Usually it's only a few percent. And often the trial has to be halted, and then we've got to analyze, and we've got tool sets now that help us understand what specifically is happening and then help the company make an assay that can test for that interaction before those folks come back into a cohort. We can then have that drug move on and have a very successful impact on the world without having to lose it in phase 2, phase 3.